Okay, hello to all of you. We are going to discuss chapter number 18 now. So, in this chapter, we are going to talk about the control of carbohydrate metabolism and how the carbohydrates, which are in the form of glycogen, how they are stored and how they are degraded. So, we have talked in the previous chapters about carbohydrates, and we have talked about the carbohydrates can exist as simple sugars, they can be monosaccharides, they can be disaccharides, or they can be polysaccharides. So, in the polysaccharides, there can be storage form of polysaccharides, or there can be structure form of polysaccharides. So, we discussed the storage form of polysaccharides include your starch, which is in plants, glycogen, which is in the animals, and we talked about cellulose, which is a structural form of polysaccharide, which is again in the plant cells. So, glycogen is a long molecule, it's a polymer, it's a polysaccharide, which means it's made of multiple monosaccharides, and the monosaccharide of which glycogen is made is glucose. And glycogen is, as I said, it's a storage form of polysaccharide in animals. So we belong to the animal kingdom, so we are animals. So in us, in our liver and in our muscles, the excess sugar is stored in the form of glycogen. So then how does glycogen, when the body needs, so basically all the I usually give one simple example here, like if you eat a lot of candy one day, so you have a lot of sugar in your system, your body doesn't need that, so your body is going to convert that sugar into glycogen, and it is going to store that into your liver, and the basically in the liver, and also some of the glycogen is stored as in the muscles. And whenever your body needs energy, then this glycogen will be broken into sugar, and then sugar can get into glycolysis, and it can release energy. So we have already studied what glycolysis is. So basically, as I said, glycogen is stored in the liver as well as muscles. In the muscles, this glycogen can be converted into glucose 6 phosphate, and glucose 6 phosphate can directly enter into glycolysis. But in the liver, the glycogen first of all breaks into glycogen 6 phosphate, and then it is hydrolyzed to glucose. So how in the liver glycogen is converted into glucose 6 phosphate and eventually into glucose? So first of all, it is converted into glucose 1 phosphate. Okay, so glycogen is first of all converted into glucose 1 phosphate, and then glucose 1 phosphate is converted into glucose 6 phosphate. So how does this happen? It is all these reactions, they're catalyzed by enzymes. So the very first enzyme which is involved is glycogen phosphorylase. So each glucose residue cleaved from glycogen reacts with phosphate to give glucose 1 phosphate, and this takes place with the help of the enzyme called glycogen phosphorylase. So it helps in cleaving alpha 1,4 linkages of the glycogen. We have discussed already what they are, and then glycogen from the glycogen, glucose will be released, and it will be combined with phosphate, and it is going to form glucose 1 phosphate. And then this glucose 1 phosphate will be converted into glucose 6 phosphate with the help of another enzyme called phosphoglucomutase. And as you know that in the glycogen there are some branches also, they can be broken with the help of some debranching enzymes. And how glycogen is produced, as I said, if you have excess glucose, it's converted into glycogen. So this process is energy requiring process. So that energy comes from UTP, uridine and triphosphate. So in this, what happens is glucose 1 phosphate reacts with UTP to produce uridine, diphosphate, glucose and pyrophosphate. So pyrophosphate is 2 phosphates together, and this reaction is catalyzed by enzyme called UDP glucose pyrophosphorylase. So this is uridine diphosphate glucose. So this is your uridine, here is your uracil, this is a nitrogenous base, this is the sugar, these are 2 phosphates, and this is glucose. So this compound is uridine diphosphate glucose. So this is formed when glucose 1 phosphate reacts with UTP. And this UTP, so first of all, this um, UDPG, uridine diphosphate glucose is formed, and then this is added to the chain of glycogen for formation of alpha 1 to 4 glycosidic linkage, and that is catalyzed by another enzyme called glycogen synthetase. And there are some enzymes which will help in the formation of branches also. And how is this glycogen metabolism controlled? The enzyme glycogen phosphorylase is the main enzyme, and the glycogen metabolism is actually controlled with this enzyme. And how this enzyme is controlled? This enzyme is actually controlled by covalent modification, because this enzyme can exist in two forms. It can exist in active and inactive form. Active form is glycogen phosphorylase A, and inactive form is glycogen phosphorylase B. So the active form is a phosphorylated form, which means that phosphate groups have been attached to this enzyme. So when this enzyme glycogen phosphorylase, when this has phosphate groups attached to it, or when this enzyme is phosphorylated, this enzyme is active, and when this is non-phosphorylated, this enzyme is inactive. And there are two more enzymes which are responsible for making them active and inactive. So the enzyme which phosphorylates this enzyme, that enzyme is called phosphorylase kinase. And the enzyme which removes a phosphate group from this enzyme and makes it inactive is called phosphorylase phosphatase. So you can see here glycogen phosphorylase can exist in two forms, glycogen phosphorylase A, glycogen phosphorylase B. Glycogen phosphorylase B is inactive, it does not have phosphate groups. So this enzyme glycogen phosphorylate can become active by the addition of phosphate groups. And who is going to add the phosphate groups? It will be added by phosphorylase kinase. And then this enzyme glycogen phosphorylase A can also become, this is active glycogen phosphorylase. It can become inactive with the help of the enzyme phosphorylase phosphatase, because that can remove the phosphate groups. So this is the way this enzyme can exist in two forms, A and B form. A is active form, it is phosphorylated form. B is inactive form, it doesn't have the phosphate groups. And who makes this enzyme phosphorylated? Phosphorylase kinase. And which enzyme makes it dephosphorylated? Phosphorylase phosphatase. So you can see here this is glycogen, because you can see all these glucose linked together. So for the removal of glucose from the long chain of glycogen, the enzyme involved is glycogen phosphorylase, and when it is functioning, it is in its active form, which is its A form, and you can see the phosphate groups are attached here, and it helps in the removal of glucose 1-phosphate from the long chain. We have just studied what reaction it is, and then it is converted to glucose 6-phosphate. And there is glycogen phosphorylase is one enzyme which is involved in this process of glycogen metabolism. Another enzyme is glycogen synthetase. We just talked about how the UDP, uridine diphosphate glucose, how it is attached to the growing chain of glycogen. It is attached with the help of the enzyme glycogen synthetase, because it is the one which helps in the formation of alpha 1 to 4 glycosidic bonds. So this is a second enzyme which is involved in glycogen metabolism, and it is also this enzyme itself is also controlled by covalent modification, just like glycogen phosphorylase, as we just talked about, but it is opposite to that. So glycogen phosphorylase is active when it has a phosphate group, and it is inactive when it doesn't have the phosphate group. So glycogen phosphorylase, when it is phosphorylated, it is inactive, and when it is unphosphorylated, it is active. So this is just a summary. The next topic we need to know in this chapter is gluconeogenesis. Now gluconeogenesis is the process by which glucose is actually formed from pyruvate. So glucose can be formed from glycogen also, as we just talked about. Glycogen is stored in liver and the muscles. From there glucose can be formed. Glucose can also be formed from pyruvate. So we have already studied in glycolysis that glucose gets converted into pyruvate, and this is a catabolic process, because in this glucose converts into pyruvate and energy is released. So it's a catabolic process. But on the other hand, pyruvate can also be converted into glucose, and that is a anabolic process, which means it is a anabolic process. It is a making up process. So catabolic means breaking processes, anabolic means building processes. So gluconeogenesis is an anabolic process in which glucose is produced from pyruvate. And I'm going to discuss from this picture, and let me zoom it a little bit. Okay, so basically, as you know, glycolysis involves 10 reactions. So gluconeogenesis, which is making of glucose from pyruvate, it involves 10 steps going in the reverse direction. Out of those 10 steps, 7 steps are same, but there are 3 steps. So 7 steps are same. What I mean is they are just the reverse of glycolysis. So in glycolysis there are 10 reactions. In gluconeogenesis there are also 10 reactions. Out of those 10 reactions, 7 reactions are just the reverse of the reactions which are taking place in the glycolysis. But there are 3 reactions which takes place in different way, and I'm going to discuss those reactions. The very first reaction which is different is the conversion of pyruvate into phosphoenol pyruvate. So in glycolysis, phosphoenol pyruvate gets converted into pyruvate, and it produces ATP. So we know that what is happening in glycolysis. So pyruvate directly gets converted into phosphoenol pyruvate directly gets converted into pyruvate in glycolysis, and it produces ATP, right? And it takes place with the help of enzyme pyruvate kinase. But in gluconeogenesis, it is going in a reverse reaction, but it involves totally different reactions. So the pyruvate, first of all, is converted into oxaloacetate, and the conversion of pyruvate to oxaloacetate. So pyruvate has 3 carbons, oxaloacetate has 4 carbons. So from where this 4th carbon is coming? This 4th carbon is coming because here is the incorporation of carbon dioxide, and this step is taking place with the help of the enzyme called pyruvate carboxylase. And as you know, gluconeogenesis is an anabolic process. It needs energy. In this step, ATP is converted into ADP. Okay, here energy is required. Then oxaloacetate is converted into phosphoenol pyruvate with the help of the enzyme phosphoenol pyruvate carboxykinase. So in this, what is happening is oxaloacetate has 4 carbons, phosphoenol pyruvate has 3 carbons. So 4 carbons, 3 carbons. Where the third carbon, where the last carbon is going? 4, 3. The 4th carbon is again here getting released as carbon dioxide. Here carbon dioxide was getting incorporated. Here carbon dioxide is going again out, and here again energy is required, and GTP, guanosine triphosphate, is converted into GDP, guanosine diphosphate. This step is different in gluconeogenesis, and there are 2 more steps which are different. Here in this step, fructose 1,6-bisphosphate is converted into fructose 6-phosphate. So in this step, basically fructose 1,6-bisphosphate has 2 phosphates, and one of the phosphate will be lost. It's getting lost here, and fructose 1,6-bisphosphate is getting converted into fructose 6-phosphate. And this is taking place with the help of the enzyme called fructose 1,6-bisphosphatase. And glucose 6-phosphate in the third step, which is different, is glucose 6-phosphate. And in this step, glucose 6-phosphate is converted into glucose. So again, there is a loss of phosphate, and this is taking place with the help of the enzyme glucose 6-phosphatase. So these are the 3 steps which are different in gluconeogenesis. Oops, sorry. Okay, oops. Okay. So I'm going to skip these slides because they just talk in detail about what I just said. Now, the last thing we need to talk about is how do enzymes control the glycogen metabolism. So, what's the role of enzymes in controlling glycogen metabolism, which means that which enzymes control when your glucose level has to be decreased or when your glucose level has to be increased in your blood. So, you know that for us, it's very important to have a certain level of glucose level in our blood. If it goes up and down, then it's not a normal situation for us. So basically, there are some hormones. So we are now going to talk about how hormones control this glycogen metabolism. So one of the hormone is insulin hormone. So basically, insulin is responsible for lowering the blood sugar level. So how does it works? Okay, now I'm going to explain you through this picture, and our chapter is not on cell signaling, so I'm not going to go into the details of lot of signaling proteins involved. I'm going to give you the basic overview how insulin actually lowers the sugar level in the blood. So insulin, so basically on our cells we have some receptors. Receptors are proteins. These receptors are called insulin receptors. These insulin receptors, they are proteins. They're embedded in the membrane, and insulin, which is a hormone, insulin is secreted in the pancreas, so it's a hormone. So it belongs to endocrine system. So through the blood the insulin will come and it will bind on the receptor. Okay, and once this insulin binds on the receptor, there are various changes, there are various conformational changes takes place in the insulin receptor, and then eventually it is going to activate some signaling pathways in the cell. And there are basically, with the insulin binding to insulin receptor, there are two different kinds of signaling pathways are activated, and one of them involves the activation of basically it involves some protein kinases. It activates, so insulin receptor, when it gets activated, it is going to activate some kinases, and those kinases are going to activate some enzymes which will be responsible in making glycogen. So one of the enzyme is glycogen synthetase. So it's going to activate this enzyme, and this enzyme is going to make it's going to convert glucose into glycogen, and this is the way it is going to reduce the blood sugar level in the blood. And another pathway which is activated, another protein kinase pathway which is activated, it results in the formation of it results in the it results in the recruitment of GLUT proteins. Now what are GLUT proteins? GLUT proteins are kind of proteins. They are transport proteins, and these proteins are responsible for the transport of glucose. So because of this kinase pathway, there is a recruitment of GLUT proteins in the membrane. So these transport proteins get embedded in the membrane, and as a result of that, the sugar, the glucose which is in the blood, which is outside the cell. So this is the cell. So if there is a high glucose level in the blood outside the cell, these GLUT proteins will let the glucose come inside the cells, and this is the way it is going to decrease the blood sugar level. And another enzymes which is involved are not the enzymes but the hormones. The other kind of hormones which are involved are glucagon and epinephrine, and their function is to increase the blood sugar level. So when the blood sugar level is going down, their function is to increase the blood sugar level. Again, I'm not going to go into the very much details of the signaling. So basically these enzymes, these hormones, glucagon and epinephrine, they basically activate another enzyme called adenyl cyclase. And adenyl cyclase is an enzyme which actually breaks ATP, adenosine triphosphate, into cyclic AMP, and cyclic AMP is a very important secondary messenger in our cells, and this cyclic AMP then eventually goes and activates another enzyme called pKa. It's called cyclic AMP dependent protein kinase, and this pKa is going to activate various other signaling pathways, and they are going to help in the increased level of glucose in the blood. So this is the way these hormones, they control the glycogen metabolism. So that's all what we need to do in chapter number 18. If you have any question, please.